Hi, everyone. Welcome to BinaryBio. If you're interested in bioinformatics, computational biology, and problem solving, you're in the right place. This is the SeqSolve series where I'll be solving problems from SeqSolve. SeqSolve is an interactive platform designed specifically for bioinformaticians, often described as LeetCode for bioinformatics. It provides a hands on environment to master computational biology through a series of structured coding challenges. While platforms like Rosalind also offer bioinformatics problems, SeqSolve emphasizes the modern interview style experience, making it highly relevant for students and professionals looking to enter the biotech workforce. If you're a student, researcher, or someone preparing for bioinformatics interviews, this series is for you. So let's get started with our very first problem. The first problem we'll solve is the GC content problem, which is one of the most fundamental problems in bioinformatics. Before solving it, let's understand what GC content means. DNA sequences are composed of four nucleotides: adenine, thymine, guanine, and cytosine. GC content is simply the percentage of nucleotides in a DNA sequence that are either G or C. GC content is biologically important because GC rich regions tend to be more structurally stable and often correlate with functional regions in genomes. In this problem, we are given a single DNA sequence. Our task is straightforward. Count how many G and C bases are present in the sequence, divide that number by the total length of the sequence, multiply by 100 to calculate the GC percentage. So, this problem tests our basic string processing skills. Why does GC content matter? The primary reason GC content is discussed in the lab is stability. A and T are connected by two hydrogen bonds. G and C are connected by three hydrogen bonds. Because of that extra bond, GC rich DNA requires more energy, higher temperatures to melt or separate the two strands. This is a critical factor when scientists design PCR primers or probes. If the GC content is too high or too low, the experiment might fail. Let's look at how this data is used in a real lab setting. Species identification and classification. As shown here, when we encounter an unknown microbe sample, the GC content acts like a molecular fingerprint. Different groups of bacteria have distinct GC signatures. For instance, actinobacteria are known for being high GC, while firmicutes are often low GC. By calculating this percentage, bioinformaticians can narrow down the identity of a mysterious organism before even looking at specific gene sequences. Optimizing PCR experiments. In the lab, PCR, polymerase chain reaction, is used to amplify DNA. This requires heating the DNA to separate the strands. Because guanine and cytosine share three hydrogen bonds compared to only two for adenine and thymine, high GC sequences are stickier. As you see on this PCR machine display, we often have to reach temperatures as high as 95.0 degrees Celsius to denature these strong bonds. Knowing the GC content allows us to predict the exact melting temperature needed for a successful experiment. Overcoming sequencing challenges. Modern DNA sequencing isn't perfect. We often run into something called GC bias. As shown in this data readout, sequencers sometimes struggle to read regions that are extremely GC rich or AT rich. This results in underrepresentation of those parts of the genome in our final data. By calculating GC content programmatically, we can identify these problematic areas and adjust our computational models to account for the missing information. Here, I'll walk through the code line by line and show how we compute the GC percentage programmatically. Now, you might be tempted to just use the dot count method twice. It's short and sweet. But as bioinformaticians, we care about computational efficiency. When you call dot count G and then dot count C, Python has to walk through the entire DNA strand from start to finish two separate times. By using a simple for loop instead, we only walk through the sequence once. We check every base, and if it's a G or a C, we increment our counter. This single pass approach is much more scalable when you're dealing with gigabytes of genomic data. After running the code, we obtain the GC percentage of the given DNA sequence, which completes the problem. That's it for today's video. In upcoming videos, we'll solve more SeqSolve problems and gradually build strong bioinformatics problem solving skills. Thanks for watching. See you in the next one.